Yes, sir.
Okay, good. So let me give you a brief overview of what we were doing in last few classes, and then we can move ahead from this point where we left in the last class. So we studied till now that how the membrane potential is established across the neuronal cell membrane, especially around, on either sides of the axon, and how does it play a role in generation of what is called as an action potential, and how does the action potential traverse all throughout the length of the axon. So for understanding that, we understood the architecture of what is called as the voltage gated sodium ion channel. We also talked about the resting potential maintaining resting potential ion channels. We also talked about that when the voltage gated sodium ion channels open, how does the action potential, which is basically the axonal membrane depolarization, gets channelized to the ends of the axon in a unidirectional fashion because of the property of the voltage gated sodium ion channels of getting inactivated after a particular period of time. So they allow only some degree of sodium ions to enter, and that is enough to bring about membrane depolarization, which is channelized, no doubt, in both the direction. But in response to closing, after closing up of voltage gated sodium ion channels, behind those there are voltage gated potassium ion channels which are allowed to open, and they uh, certainly re-establish or help in the re-establishment of the membrane potential, and the membrane gets repolarized back so that it can reinitiate another action potential later on. This scheme also ensures that the the migration of the action potential always happens towards the axon terminus, and it never goes in a backward direction. To understand how the repolarization is done by voltage gated potassium ion channels, we also understood the architecture of the potassium ion channels is more or less similar to sodium ion channels. The only exception is that the inactivating segments, the number of inactivating segments, these ball-like structures that you see, are more in delayed voltage-gated potassium ion channels. They are also called as the channels which open in response to change in potential, but the opening is slightly delayed compared to what we have seen in case of instantaneously opening sodium ion channels. Now, we also addressed this question that when we look at the size of the potassium ion and compare it with the size of the sodium ion or any other positively charged ion for that matter, which exists inside the cell, then it appears that potassium ions are quite big in size compared to sodium ions or compared to protons. And because of that, there is always a chance that you might, through potassium ion channels, you might get a leakage of sodium ions as well. But the mechanism which is used by the selectivity filter of these channels that we understood, and the mechanism is actually based upon the hydration shell around the sodium and potassium ions. Because sodium ions are smaller, they will require less number of water molecules to hydrate, and because potassium ions are bigger in size, they will require more number of water molecules to hydrate. And as soon as these hydrated ions approach the vestibule, or you can say the channel uh, inner core, these water molecules are stripped off from the ion, and then the ion is made to interact with the oxygens of those carboxylic acid groups which are positioned in a manner that they orient exactly in the manner how the water molecules are associated with potassium ions. So if we compare potassium ion interaction with that of sodium ions, then it appears like this, that the potassium ions are actually going to be uh, associated with water molecules which are pretty close enough. Okay, so you can look at this interaction of the oxygen of water with potassium is having a very close association with potassium. And when you compare this association with the oxygens that are lining the core, these are those oxygen residues. I hope that you can see my cursor. Okay, so these are those oxygen residues which I'm talking about. This one and this one. And they are oriented exactly in the same manner how the oxygen molecules of water are oriented when water hydrates potassium ions. Now, when you compare this with that of sodium ion, now this is just for comparison, okay? The number of water molecules that are going to associate with sodium ion is lesser compared to that of potassium ion. But when you compare that in general with potassium ion interaction, now because sodium ion will be smaller in size, these oxygen will be slightly closely placed compared to what they were placed in case of potassium ion hydration. Now, if that is so, then the core where the potassium ion channels are going to be held by the potassium ion channels, that area will never be having a very good kind of an interaction with sodium ions, because these oxygens you can see, these are of those carboxylic acid groups and they are quite far apart here compared to the oxygens interacting with potassium ions. So what happens is that these sodium ions don't get completely stripped off from the water molecules. And because they are not going to get completely stripped off from the water molecules, some water molecules still remain interacting with sodium. And that's what makes a sodium ion more bulky than potassium ions to get inside the vestibule. I hope that the point I'm making is understandable to everyone. Is it?
So can you please repeat?
Which portion do you want me to repeat?
So last one.
The point is this, that obviously when we are talking about interaction of potassium with oxygen of water and sodium with oxygen of water, the water molecules orientation around sodium and water molecules orientation around potassium will be different because of their sizes, their individual sizes. And because this is different, because this orientation and this orientation, although it looks similar here, you can imagine that if I somehow freeze these oxygens, imagine that I have frozen these positions of oxygen. So that what will happen is that when potassium ion is approaching the vestibule, that means is approaching this channel, then these water molecules can be stripped off. Stripped off in the sense they can be removed, because potassium ion can now interact with oxygens of the carboxyl group which are lining the vestibule. Is this much fine?
Yes, sir.
Now imagine that instead of potassium, you have sodium, which will be smaller in size compared to potassium. Now, when this hydrated molecule of sodium ion approaches the same vestibule of potassium ion channel, what will happen is that these water molecules cannot be completely stripped off. The reason is sodium can interact at the same point of time only with two frozen oxygens of the vestibule lining, right? The other two frozen oxygens here cannot interact, and hence this sodium ion will continue to interact with the oxygens of water that it is holding for hydration. Now, when I'm saying this, then these two water molecules will make it much more bulky for this sodium ion to pass through this vestibule. Are you getting the point?
Yes, sir.
and hence sodium ions will not be allowed to enter. I hope that you have got the point. Okay,
Yes, sir.
so that makes potassium ion channels more selective for potassium ions rather than for sodium ions. I hope that the point is clear.
Yes, sir.
Can you see this now? Okay, good. So, so that is how the understanding is. So, if I want to go ahead from this point, okay. So the understanding could be like that potassium ion channels will become much more selective or much more sensitive for allowing only the movement of potassium ions across the membrane. Now, for time being, we have understood that if I want to draw, say suppose this is how the axon looks like, and this is the cell body portion. Okay, say suppose the nucleus, and this is the axon terminal end, where the axon seems to have these these portions which end up into small bulbs. Looks like this. I'm not drawing it completely just for our understanding for time being. Okay, they are branched up into several large areas, but for time being, let us keep it simple. So now, if this is the dendrite portion, so there will be more branching here and so on and so forth. We'll keep it simple for time being. So say suppose this is the neuronal cell. So now I have drawn it in a slightly opposite orientation. But just imagine that the action potential begins from here. Okay, this is the site where the action potential begins. Okay, this is the portion which is called as an axon hillock. Okay, and here is the action potential going to originate. Now, when it originates from here, okay, now how it happens, that also we are going to discuss, but not now. We'll first need to understand that how it passes towards the end of the axon. The understanding goes like that that you have opening up of sodium ion channels here. The sodium ion channels I will draw like this. Potassium ion channels I will draw like this. Okay, just for making the difference. So sodium ion channels will open, and there will be several such sodium ion channels that will continue to open all throughout the length of the axon. And then there is re-establishment and all that we have talked about. So now the question arises that okay, this traverses all throughout towards the end of the axon. So there will be some sodium ion channels here also, here also, here also, and so on and so forth. What happens exactly now? Okay, so the action potential traverses like this. Action potential may go along with all the axon terminals. But what happens at the axon terminals, or how this action potential is going to channelize the information to the next neuron? So, does anybody have any idea regarding that? What happens here? Yes.
So the action potential moves with the help of synapse and then enters the new neuron.
So, do you want to say that the action potential will continue, and there is a synapse here in this area, right?
Yes, sir.
So just for understanding, imagine that this is a neuron, and this is the dendrite of the neuron, and this portion of the dendrite is forming a synapse. Just for our understanding, okay. of another neuron whose axon terminal is here. It looks like this. This is the bulb. This is the synapse area, right? So
Yes, sir.
is this good? So if we are talking about this portion, this becomes the synapse of one neuron. So say suppose the action potential arrives here. So how will it get channelized in this dendrite then, and through this neuron?
Sir,
Yes.
there must be something which accepts that action
Okay.
potential.
So you want to say that if one neuron has fired an action potential, then it is necessary that the next neuron should also fire an action potential. Is this what you are trying to say? So say suppose this neuron has fired an action potential. It reaches to the axon terminal, and now this neuron has to fire another action potential. Is this mandatory?
Sir, I'm not sure about this.
Okay, so how come it is decided that whether the next neuron should fire an action potential or will not fire an action potential, or how it will respond to the previous neuron? So there is some nomenclature that is given here. This is called as the presynaptic neuron. It is, say, for example, neuron number 1, which is in connection with another neuron through this synapse, and then the other neuron continues. So this is called as the postsynaptic neuron. And when we look at these membranes, then they are called as presynaptic membrane and postsynaptic membrane. So now, okay, you have learned something about neuromuscular junction, if I am not wrong. Have you? During muscle contraction? Yes.
Yes, sir.
Okay. So what is it? And can anybody let me know that? Do you remember that when a neuron is stimulated, in order to cause or relay that stimulation to the muscle to undergo contraction, what exactly happens? That will help you in remembering this portion as well, that what exactly happens at the axon terminal. Yes. What
Sir,
happens?
neurotransmitters are released.
Okay, but muscles, do they understand the language of neurotransmitters? Yes. Is it a valid question to ask?
Yes, sir.
Yes. So, will muscle understand the language of neurotransmitters, according to you? Yes or no? Just yes or no. We'll talk about the details later.
Yes, sir.
They should understand. That's what you're trying to say,
Yes,
Akshay?
sir.
Okay. So, are they going to behave in the same fashion how the neuron behaves after learning the presence of neurotransmitter?
Sir, there is a different mechanism
Okay.
inside means inside muscles.
Okay. What is that mechanism that you have learned, I believe?
Sir, presently I am not able to recall it,
Recall.
but
Okay, fine. Fine, no issues. Okay, but have you heard about this compound?
acetylcholine.
Yes. So what do you think? What is this? Is it a neurotransmitter? Is it a
Yes, sir.
hormone?
Yes, sir.
It's a neurotransmitter.
It's a neurotransmitter. And how does the muscle membrane reacts with acetylcholine?
Sir, there are receptors.
Receptors for acetylcholine. And what happens when acetylcholine binds to those receptors? Yes.
So, pardon, please.
What happens when acetylcholine binds to the receptors present on the muscle cell? Okay. Okay. Can anybody guess where this acetylcholine must be manufactured? Where is it synthesized? Because it's a neurotransmitter.
The cell body,
A cell body of what?
neuron.
Obviously. Because it's a neurotransmitter, the cell body of this neuron should say, suppose, manufacture acetylcholine, right? And acetylcholine, you believe, is released where? So acetylcholine, I will say that it is present here. Where is it released? In order to bring about muscle contraction, where do you think acetylcholine must be getting released? Obviously at the neuromuscular junction, right? So say suppose like synapse, there is a junction which is very similar to synapse, but this is a curvature which is of the cell membrane of a muscle. Okay, wherein these axon terminals are actually getting embedded. So acetylcholine will be released here in this space, right?
Yes, sir.
Right. So now first question. We'll come back to the question that I was asking. But first question is, acetylcholine is getting manufactured here, right? And is getting released here. It also needs to travel all throughout the length of the axon and get down, correct?
Yes, sir.
Right. So how come it goes to the terminal? This is question, another question, right? So there are a few things that we need to address here. First is, how is one neuron communicating with another neuron? So when we were talking about this communication, we landed up into understanding that the neurons are known to release what are called as neurotransmitters, right? And one such neurotransmitters you have already learned about, and that is acetylcholine. Acetylcholine somehow is going to stimulate the muscle cell to undergo contraction, correct?
Yes, sir.
Right. So what it should be doing in order to bring about muscle contraction? Now, let's get back to your basics of muscle contraction. Which of the ions are actually involved in bringing about contraction of the muscle?
Calcium.
Calcium. Okay. And do you think by any chance acetylcholine has to do something with increasing the level of calcium inside the muscle cell?
Yes, sir.
Okay. So we are getting to uh, what is happening inside the muscle cell, right? Okay. So now say suppose acetylcholine binds to its receptor and somehow it is responsible for opening up of What? I don't know whether you have learned about this or not. Have you ever heard about something called as a T tubule? Yes?
No, sir.
No. Okay. Okay. T tubule is a kind of a membranous channel which runs all throughout the muscle cell. Okay. Uh, and it is actually going to channelize whatever that is sensed by the acetylcholine at the site of the neuromuscular junction. Okay. Now, why we are talking about T tubule is because T tubule is something like this, runs all throughout the muscle cell fiber, and its membranous structure is in close contact with the membrane of what is called as, have you ever heard about sarcoplasmic reticulum?
Yes, sir.
Yes or no?
Yes, sir.
Okay, what's the function of the sarcoplasmic reticulum? Come on, I'm asking just the things what you have already learned about. Simple ones. Yes, no idea?
Sir, it is a storehouse of calcium ions.
Yes, it is just a storehouse of calcium ions. Okay, so now what happens? What must be happening? You can guess it now. Okay, sarcoplasmic reticular membrane is having, say, it is something like this. Imagine that this is a sarcoplasmic reticulum. So it will be full of calcium ions, right? It is in close contact with the T tubule running through. So whatever that is channelized through the membrane of the T tubule will be channelized through the membrane of sarcoplasmic reticulum as well. That is responsible for opening up of the channels which are going to allow a quick rush of calcium ions outside the sarcoplasmic reticulum. Do you remember this mechanism? Yes or no? Okay. Okay, what I will do is that I will break the ice. Okay, what does the calcium ions do here? Basically, if I increase the level of calcium ions inside the sarco sarcomere, what exactly happens? Okay, so you will understand why I am focusing more on the muscles. Okay, okay. So I don't think that you're you're trying to you're getting any thoughts about what calcium ion is going to do. It's basically very interesting mechanism. So in the sarcomere, there are thick and thin filaments, right? Arrangement is somewhat like thick filaments are like this. There are thin filaments. Sorry, there are thin filaments like this. And then there are thick filaments alternating. Correct? Remember, I'm just drawing a rough figure. Okay? What happens is outside from thick filament, there are the bulbs which originate, which are in contact with the thin filament, right? And this is made up of a protein which is called as myosin. Have you heard about this?
Yes, sir.
Okay.
Yes, sir.
And what is the property of myosin? Okay, just for your uh, your remembrance, myosin is a motor protein which is there inside the muscle cells and which is responsible for the movement or the muscle contraction. How it happens is quite interesting. This, with which it is in contact with, this is the actin filament. And actins, if you remember, actins are going to form the thin filaments where they are made up of structures. This is more precisely called as F-actin. And the property of myosin is that it is an actin activated motor protein. Okay, so it has a head group which looks somewhat like this. Okay, which is in contact with the F-actin filament, which is drawn like that. Okay. Now the situation is that before it forms a contact, so unless it forms a contact, it cannot. A myosin head has a capability of hydrolyzing ADP, and this generates what is called as the power stroke, by which it bends. It bends in the sense that previously, if it is here, after ATP hydrolysis, it comes out to be, the location changes, it goes to here, this position. Okay, to bring about this ATP hydrolysis, it must form a contact with actin. And this actin is actually enveloped by another protein, which is called as tropomyosin. Have you heard about this?
Yes, sir.
Yes, sir.
And tropomyosin is in close contact with troponin. Have you heard about this?
Yes, sir.
Yes, sir.
Okay. So now can you tell me, because you have heard about all these proteins, can you tell me what's the role of calcium?
Sir,
Sir,
it will,
calcium binds
Yes.
to the troponin C. It is a site where calcium attach.
Okay, so what happens then?
Then it changes the structure of the particular molecule, and hence there is power stroke.
So see, power stroke is generated by myosin, right? You're
Yes, sir.
talking about interaction of calcium with troponin, right?
Yes, sir.
So what's the relation? Do you think this is a valid question to ask?
Yes, sir.
Right. So what's the relation? If troponin is binding to calcium, how myosin is unable to bring about the power stroke through ATP hydrolysis? You have learned this. Okay. All three of them are in very close contact with each other. So when one changes the conformation due to calcium ion binding, the other also changes the conformation. And because of that, the position where myosin head is in close contact, rather, it is not forming a contact. Just imagine. Now, just for our understanding, if these are the actin residues, tropomyosin is oriented somewhat like this. And troponins are in very close association here somewhere. Okay. Now, when they bind to calcium, what happens is that this position changes. And because of it, it gives a slight space for the myosin head. This is the myosin head to make an entry and interact with actin. And as I have told you just now, it is an actin activated kind of an ATPase that it has, this ATP hydrolyzing activity. So unless it interacts with actin, it cannot hydrolyze ATP. Are you getting the point? Yes. So it is a cascade that is happening. Calcium, when it is raised, it binds to troponin. Troponin is in close contact with tropomyosin, and tropomyosin, when it's changing its position, it is actually opening the gates for an axis of myosin head to interact with actin. And as soon as myosin finds a contact with actin, it hydrolyzes ATP. Interesting?
Yes, sir.
And that generates a power stroke, which actually brings up the contraction. Okay, I am not here to teach you what is muscle contraction, but just for revision. So that is happening in response to acetylcholine, right? So acetylcholine must be doing something here so that the sarcoplasmic reticular membrane understands that I should release calcium ions. And what it is doing is that it is actually binding to certain receptors which are very similar to the receptors which are present in the postsynaptic neuronal membrane. And these receptors are of two types. Okay. One receptor is of the type. Now, just you will tell me now. Okay. Imagine this. Like one receptor looks like this across the membrane. Okay. And it has a property that it binds to acetylcholine, and in response to binding, there is opening up of a channel. And if the channel that it opens is, if the channel that it opens after binding of acetylcholine, if the channel is of sodium ion, what will happen to the postsynaptic neuron? Is the question clear? Should I repeat?
Yes, sir.
Okay, now imagine that this receptor to which acetylcholine binds, this receptor is a kind of a channel. Okay, but the channel opens in response to binding of acetylcholine. So now if I give you this, that this receptor channel that we are talking about is permeable to sodium ions, what will happen to the postsynaptic neuron?
Depolarization.
Right. So will this depolarization be channelized now all throughout the next neuron?
Yes, sir.
Right. So now if I excite the first neuron, that means I cause a depolarization here. And if you have a kind of a channel in the postsynaptic membrane, which responds to the neurotransmitter acetylcholine in the same manner by opening up of sodium ion channels, then this kind of a relay will be referred to as, that means these kind of receptors for acetylcholine are nothing but ligand gated receptors. Ligand is nothing but acetylcholine. Okay, so I can call them as receptors of acetylcholine, or for understanding and for better remembrance, I can call them ligand gated sodium ion channels. Yes or no?
Yes, sir.
Yes, sir.
So now interesting thing that is happening at the synapse is that as soon as the first neuron releases acetylcholine here, then it is going to excite the next neuron, because the next neuron has a acetylcholine binding sodium ion channel, or acetylcholine gated sodium ion channel. Interesting. Now the
Yes, sir.
now the thing that we need to uh, focus our attention to is how is acetylcholine being transported all throughout the length of the axon and is going to be released here, will it be a quick process or will it be a very slow process? According to you? Because acetylcholine is getting manufactured at the cell body, right?
Yes, sir.
So now if I want to transfer acetylcholine gated neuronal message, then I will have to transport acetylcholine all throughout the length of the neuron and then release here, correct?
Yes, sir.
Okay. So will it be a fast process or slow process according to you?
Sir, slow.
Slow process, right? But you want this muscle to contract fast, or you want this neuron to carry the message fast. What will you do then? Do you think this is a valid question to ask?
Yes, sir.
Okay, so what you will do? Your view. I just want your view in this. What is done by the neuron, I will tell you, no doubt. But what you can do? You can have two ways of doing it. Okay. One is what we have talked about through a neurotransmitter. And another way is by forming a synapse, which is called as, now this kind of a synapse where there is a release of neurotransmitter, the neurotransmitter binds to the postsynaptic neuronal membrane and then initiates an action potential. This type of a synapse is called as, is grouped under the category of chemical synapses. There is another synapse which is called as an electrical synapse. What according to you must be different here? The synapses in the electrical synapse are held so tightly, just like this. The T-tubule and the sarcoplasmic reticulum are held so tightly in the muscle cell, right? In the similar fashion, if I hold these two membranes so very close, then what will happen? As the action potential arrives here, it will immediately get transferred directly to the next membrane, will it not?
Yes, sir.
That type
Yes,
of
sir.
a transmission is called as the electrical synapse. Okay, this is very fast. This electrical synapses conduct very, very fast. We'll talk about that, where these are normally functional. Chemical synapses compared to electrical synapses slightly slower, but it's only slightly slower. That works for us, right? Because if we want to move a muscle, how much time do you take to bend your arm? Within milliseconds you can do it, right? Okay, and that is the speed at which even chemical synapses work. So you can imagine what must be the speed of electrical synapses, right? So one option I have given you, you can excite the next neuron by electrical synapses. Okay, but if you want to work with chemical synapse, your neuron needs to work harder, because it needs to synthesize the chemical, that is the neurotransmitter, and then it needs to transport that neurotransmitter to the axon terminal, and that is done by means of certain small packets of neurotransmitters. And these packets are called as the synaptic vesicles. Synaptic vesicles are the ones which are just the packets of the neurotransmitter. We'll consider for simplicity's sake acetylcholine. So they get filled up with acetylcholine, and then these are transported to the exon terminal. Interestingly, this transport to the exon terminal is done by myosin. But this myosin, which is doing the transport, is a different type. This myosin is of different type, and the myosin which is there inside the neurons, which is going to transfer the synaptic vesicles to the axonal terminal, is called as myosin 5. And sorry, the interesting thing about this, just forget the spelling, okay? Interesting thing about this myosin 5 is it looks like this. Now we know that the myosin has a head, right? And we have I have just told you that the head is an actin activated ATPase. You remember that? I hope so. So now imagine this, that you have a myosin which looks like this. There is a myosin head, and there are two myosin heads, right? If you remember the structure of myosin, there are two myosin heads which are very close to each other. Now what happens is this. If there is a synaptic vesicle, the myosin heads protrude out from the synaptic vesicles, and they look like this. Sorry. So one myosin head, another myosin head. They look like this. Okay. And interestingly, if you remember, when we were talking about the architecture of the neuronal cell, I told you that there is a cytoskeletal network that maintains the cell's architecture, right? You remember that?
Yes, sir.
Okay. It's called a cytoskeleton. And the cytoskeleton is containing actin in it. So I can say this, that there are tracks of actin inside the neuronal cell, and these myosin heads, as they come in contact with actin, you know what happens when myosin head comes in contact with actin. It generates a power stroke, right? So what will happen? Yes? It will actually propel. Now it actually walks along the tracks of actin, and this cargo is actually deposited at the exon terminal. So it gets concentrated here. Interesting. This kind of a transport from the cell body to the exon terminals is called as an anterograde transport. Okay. Is there any doubt in this?
So can you please repeat it?
What do you want me to repeat? Do
So
you want
last
me
part,
to?
actually I'm having network problems, so please.
The idea is like this, that the myosin heads, as I have just told you, myosin heads are actin dependent ATPases. They are going to generate the power stroke only when they are in contact with actin. So imagine that you have a synaptic vesicle which has a myosin protruding out of it, and the myosin forms a contact with the actin cytoskeletal network that is here. Okay. What will happen as soon as it forms a contact? It will start to hydrolyze ATP that will generate a power stroke, right? And that will bend the myosins. As they bend over, there will be other myosins which will be protruding out here. They will form a contact. This will lose and this will continue to move, right?
Yes, sir.
And this actually is responsible for the transport. Now this, I have given you a very short part of the story, okay? There are other proteins also involved in this process which are associated with uh, kinesin and dynein. These are other motor proteins that are also there, and they are microtubule driven. So it is similar to this, but I don't want to go into the details of it for time being, because that will just complicate the matters for you. Microtubule is a much higher version of cytoskeletal network. So in short, there is a cytoskeletal track by some or the other protein, and there are some motor proteins protruding out from the synaptic vesicle. When they form the contact, that propels them towards the axon terminal. This kind of a transport is called as an anterograde transport. Is this much fine? Do you have any doubts in this? Yes?
No, sir.
Okay, good. So what I will do is that this is the way, okay, now we have solved two questions here. This is the way that the neuronal cell synthesizes the neurotransmitter at the cell body and transports the neurotransmitter to the end. Okay. And then it can release it at the end when the action potential arrives here. Now, the question that we need to address in the next class is that how does the synaptic vesicles release the neurotransmitter in the synaptic cleft, or what guides them to release the neurotransmitter into the synaptic cleft? And there is one more question that actually you should address, but I will not address it right now, or I will not give it to you right now. It's a different question altogether. Okay, we'll talk about that when we understand that how the neurotransmitter is released into the synaptic cleft. When all the synaptic vesicles have already concentrated here, then if the neuron needs to fire, one thing you need to understand, if the neuron needs to fire, it already needs to have all these synaptic vesicles concentrated at the axon terminals, right? Okay. So tomorrow's class will be focusing on that how the neurotransmitter is released in the synaptic cleft when the action potential arrives. It should be released only in response to action potential, right? It should not get released over and over again. Then there is no fun in generating an action potential. I hope that the point is clear.
Yes, sir.
Yes.
Okay. So if you don't have any questions, then I will call it a day for today and we will end the class. Or if you have any questions, then